Hello everyone, and welcome to Pearson Bio Academy. I am Fazna Imran.
This is the place where you can top up your biology knowledge with our past paper explanation through very short and clear video series. Let's get diving. And today we'll be focusing on Pearson Edexcel International Advanced Level, paper reference WBI 1101, Unit 1, which covers molecules, diet, transport and health. Before we dive into the sessions, let's start with a brief introduction. I'll outline the key topics that are covered in Unit 1, and these topics are directly derived from the textbook, and they include chemistry of life, mammalian transport system, cardiovascular health and risk, membrane and transport, proteins and DNA, gene expression and genetics. So all the questions we'll be discussing will come from these 6 main areas. To make it super simple, I have made some further categorization according to a question paper, which is really helpful to understand things better. They include carbohydrates, chemistry of water, proteins, lipids, and finally enzymes. However, you might learn enzymes under topic 2, but here I have brought it under chemistry of life because most of the time these questions are appearing together. Out of those topics, today we will be focusing on carbohydrates, questions from 2022-2024. Before we start tackling the questions, there are a few key facts about carbohydrates that you should know. Don't worry, I will not repeat the theory over and over, just the essential that you will need to answer the questions. In this slide, we'll focus on 3 main categories of carbohydrates. They are monosaccharides, disaccharides, and polysaccharides. First, let's talk about monosaccharides, and we'll cover 3 important ones: GGF, galactose, glucose, and fructose. So if you ever find it difficult to remember, just think of GGF as a simple way to recall them. Next are disaccharides. There are many types, but we'll focus on 3: sucrose, maltose, and lactose. And finally, for polysaccharides, while there are several types, we'll concentrate on starch and glycogen in this session, and we will explore about cellulose more when we get to topic 2. If there is only one single unit, then they are known as monosaccharides. And if there are 2 monosaccharides joined together by a glycosidic bond, then they are known as disaccharides. And if there are more than 3 units, but lesser than 10 units, then they will be known as oligosaccharides. Glucose is a common example of a monosaccharide. This image taken from your student book shows that glucose can be in 2 isomeric forms, alpha glucose and beta glucose. You can observe the difference between them, specifically the position of hydrogen and hydroxyl group. In alpha glucose, the hydrogen is positioned above the ring, while the hydroxyl group is below. In beta glucose, the positions are reversed. So far, there haven't been any questions specifically about beta glucose, mainly because we are focusing on starch and glycogen, which are both related to alpha glucose. However, it's still important to be prepared. In the future, you might be asked to draw alpha glucose, so make sure you practice this. Also, be ready to differentiate between alpha and beta forms, as this understanding could be useful. I can say this is the mantra of carbohydrates. There are many questions that repeatedly test your understanding of how specific disaccharides are formed. When two glucose molecules combine, they form maltose. Glucose pairs with fructose, it produces sucrose. When glucose joins with galactose, they form lactose. So make sure you are familiar with these combinations as they are key concepts that often come up in exams. First, let's take a look at some questions related to the formation of these disaccharides. The first question is from the January 2020 paper. Which of the following are monosaccharides present in lactose? So if you are familiar with lactose, this question is straightforward. The monosaccharides that make up lactose are galactose and glucose. The key idea is here to remember that glucose is one of the main monosaccharides and the other one paired with it will be different. In the case of lactose, it's galactose. You can even see the hint in the name itself. Lactose contains lact, which can help you to remember that it's made up of galactose. So the correct answer is galactose and glucose. The next question is from the January 2021 paper and it's once again about lactose. The statement reads, people with lactose intolerance cannot digest lactose, and lactose intolerance is due to a lack of the enzyme lactase. The question asks, which are the products of lactose digestion? This is a straightforward question and it's similar to the one we just discussed. So when lactose is digested, it breaks down into galactose and glucose. So even though the questions may vary, the concept remains the same. The correct answer here is also galactose and glucose. The next question is from the June 2021 paper. The statement reads, blood glucose levels can become high following the digestion of carbohydrates. And the question is, which of the following can be digested to release glucose? To answer this, remember that to release glucose, the compound must be a disaccharide or a polysaccharide. Let's go through the options. Option A, fructose and sucrose. So fructose is a monosaccharide, so it cannot release glucose on its own. Sucrose, however, is a disaccharide made of one fructose and one glucose molecule. Since fructose can't release glucose, option A is incorrect. Option B, both fructose and galactose. So both fructose and galactose are monosaccharides, so neither can release glucose. Therefore, option B is also incorrect. Option C, galactose and lactose. Galactose is a monosaccharide, so it doesn't release glucose. However, lactose is a disaccharide which made up of one glucose and one galactose molecule, so it can release glucose when digested. But since galactose doesn't fit, option C is not the right answer. Option D, lactose and sucrose. Both lactose and sucrose are disaccharide. When lactose is digested, it releases one glucose and one galactose molecule. Similarly, sucrose releases one glucose and one fructose molecule. Therefore, both can release glucose. So, the correct answer is answer T, both lactose and sucrose. Additionally, you have to keep remember that maltose can also release glucose as it's made of two glucose molecules. Next, let's talk about glycosidic bonds. A glycosidic bond is a type of covalent bond found in carbohydrates. It's responsible for linking sugar molecules together to form disaccharides, oligosaccharides and polysaccharides. For instance, when forming disaccharides, two monosaccharides such as glucose molecules are joined by a glycosidic bond. This bond forms through a reaction that removes a water molecule known as a condensation reaction. Now let's look at how a condensation reaction occurs. Initially, you will notice that a water molecule is produced as part of this reaction. Following this, a glycosidic bond forms between the two monosaccharides. And this specific reaction is termed a condensation reaction because it results in the joining of two molecules with the release of one water molecule. The bond formed here is known as 1,4-glycosidic bond. This is because the bond occurs between the first carbon of one monosaccharide and the fourth carbon of another. In recent past papers, examiners have been focusing more on students' understanding of how this reaction occurs and how the bond forms. So it's important to be clear on this concept. I have included a diagram from your student book because, as you know, exam questions often refer to images provided in your textbook. This diagram illustrates how a condensation reaction forms. Although the carbons are not labeled here, the positions are implied 1 through 6, showing the carbon structure. In this example, two alpha glucose molecules are involved. You can see that the hydroxyl group from the first carbon of one glucose and the hydrogen from the fourth carbon of other glucoses are removed to forming a water molecule. So this is how a 1,4-glycosidic bond is created between the two glucose molecules. So when you are asked to draw or complete a diagram of condensation, make sure to illustrate how the H2O molecule is released and clearly label the 1,4-glycosidic bond. Now let's discuss the hydrolysis reaction. So in a condensation reaction, a water molecule is removed to form a bond. However, during hydrolysis, the process is reversed. A disaccharide, which is a molecule made up of two monosaccharides, is split into two by adding a water molecule. Here how it works. We add one H2O molecule and specific enzymes, which we will cover later, facilitates this reaction. The water molecule breaks the bond with the hydroxyl group attaching to one monosaccharide and the hydrogen attaching to the other. So this is how a hydrolysis splits the molecule too. If you look at the overall process, you will notice it's a reversible reaction. In the first case, two monosaccharides join together to form a disaccharide, releasing a water molecule in the process. And this is called condensation. And conversely, when you add a water molecule to a disaccharide, it breaks apart into two separate monosaccharides, and this process is known as hydrolysis. So this is the first question from January 2021 paper where you are asked to fill out a table. The focus is on polymers and specifically polysaccharides. So you need to identify the elements present in the monomers. For polysaccharides, the monomers are monosaccharides. The question has already given the bond type, so you don't need to fill the part. Instead, you just need to write down the elements in the monomers. So monosaccharides consist of carbon, hydrogen, and oxygen. This question is from the October 2021 paper. As you'll notice, the question provides some background information first. So typically when you encounter such questions, they introduce new concepts before getting to the actual questions. Don't let this additional information confuse you, as the questions themselves are often straightforward. For example, in this question, they have mentioned that a butterfly is an insect that feeds on nectar produced by flowers. and it includes a photograph of a butterfly feeding on a flower. And the question part is here. The nectar in flowers contain nutrients including sugars, amino acids and lipids. The sugars in the nectar are fructose, glucose and sucrose. And this is the part which we have to focus more. The question, which of these contain glycosidic bonds? To determine which of these contains glycosidic bonds, remember that a glycosidic bond can only form between two monosaccharides when they combine to create a disaccharide or a polysaccharide. So in this case, fructose and glucose are both monosaccharides and do not contain glycosidic bonds on their own. However, sucrose is a disaccharide formed from the combination of glucose and fructose. So, meaning it does contain a glycosidic bond. So, the correct answer is B, sucrose only. This question is from the January 2022 paper. It starts with an interesting piece of information about saffron. So saffron is a spice that is derived from parts of the flowers of the saffron crocus, and saffron contains a disaccharide called a gentiobias, which is a new term for you. However, the question focuses on more commonly known disaccharides. Read through the following description of some disaccharides, and you have to complete the description by writing the most appropriate word on the dotted lines. Disaccharides consist of two monosaccharides joined together by a dash covalent bond, and you know it is a glycosidic bond. Sucrose and lactose are both disaccharides. They both contain a molecule of glucose. Sucrose also contains one molecule of fructose, and lactose also contains one molecule of galactose. So while the paragraph, the question begins with gentiobias, but our focus is on sucrose and lactose. So the final answers are glycosidic, glucose, fructose, and finally galactose. Second part from that question, gentiobiose is formed from two identical monosaccharides, and you have to write the type of reaction. Please note that's not the type of bond, the type of reaction. Whatever the monosaccharides can be, but if they want to join together, they have to go through the reaction called condensation. The third part from the same question, you are given with the molecular mass of each of the monosaccharides in gentiobiose is 180. It's about the monosaccharides molecular mass. And also you have given with a table shows the molecular mass of the elements present in the monosaccharides. So carbon is 12, hydrogen is 1, and for oxygen it's 16. And the question is, which is the molecular mass of gentiobiose? We can approach this question in two different methods. The first one, if you know the common formula for a disaccharide, then we can do a simple calculation to find out the molecular mass. And here they have given the molecular mass of each element. For carbon it's 12. So 12 times 12 plus 1 into 22 times, and for oxygen it's 16. So 16 into 11 times, and final answer is 342. And the second method, if you can remember this reaction, you can do this in another way. So, you know, if there is a disaccharide is being formed, two monosaccharide will be joined together by releasing one H2O molecule. In your question, they have given you the molecular mass of each monosaccharide, and it's 180. So 2 times 180, and you have to subtract a molecular mass of H2O from it, and it's 18, and finally you will get the answer. It's 342. So the final answer is 342. The next question is from the May 2023 paper focusing on sucrose. So sucrose is a disaccharide made from glucose and fructose, and glucose is joined to fructose by a 1,2 glycosidic bond. Unlike other disaccharides that might involve a 1,4 glycosidic bond, here we specifically have a 1,2 glycosidic bond, so there is no need to get confused. The concept is straightforward. You are required to draw a bond between the first carbon of the glucose molecule and the second carbon of the fructose molecule. And the diagram clearly highlights these positions, showing the connection between the two carbons. And when drawing this bond, you have to remember there is no need to illustrate the formation of water molecule. You can see that the hydroxyl group from the first carbon of glucose and the hydrogen atom from the second carbon of fructose are missing, indicating that water has already been removed. Therefore, it's sufficient to just show the glycosidic bond connecting the two sugars, and you can show it like this. Second part from that question, complete the formula equation for the reaction that makes sucrose from glucose and fructose. It's very easy and we have learned already. It's a formation of disaccharide plus and formation of water. It's very easy. In the next part of the question, we have a practical experiment to determine the concentration of sucrose in a solution. And this chart outlines the steps involved in this process. We will look step by step. The first step is to break down sucrose into its components, glucose and fructose, using an enzyme. As I mentioned earlier, an enzyme can split a disaccharide into two monosaccharides. And the next step, after the sucrose has been broken down, you add a chemical reagent that specifically binds to fructose and causes a color change. It's important to remember that this chemical only interacts with the fructose molecule, not with the glucose. And step 3, finally, you measure the color intensity of the solution. And here the color intensity indicates the concentration of sucrose as it reflects the amounts of fructose present after the enzymatic breakdown. Let's go through a schematic representation of the test. Initially, you have a colorless sucrose solution which contains glucose and fructose linked together as a disaccharide. And when you add the enzyme, it breaks down the sucrose into its individual components, glucose and fructose molecules. Now the solution contains these monosaccharides separately. Next, you add a chemical reagent that specifically binds to fructose, causing a color change. The solution will develop a color due to this interaction between the reagent and the fructose molecules, and the color intensity depends on the amount of fructose present, which in turn reflects the original concentration of sucrose in the solution. Now let's examine the experiment a bit more closely. For sucrose, which is a disaccharide, it's break down into one glucose molecule and one fructose molecule. After adding a chemical, the fructose will cause a color change. Let's consider some calculation to understand this better. Suppose you add 0.1 m sucrose to the solution. Since sucrose splits into one glucose and one fructose, this will yield 0.1 m glucose and 0.1 m fructose. However, we are only focusing on the fructose as it's the one that reacts with the chemical to produce a color. Therefore, 0.1 m sucrose will result in 0.1 m colored fructose, and the color intensity will correspond to this concentration. And if you add 0.2 m sucrose, it will produce 0.2 m fructose, leading to 0.2 m of the colored product. In this case, the color intensity will be double that of the 0.1 m solution. And similarly, for 0.3 m sucrose, it will produce 0.3 m colored fructose, and so on. So from this, we can deduce that the concentration of sucrose is directly proportional to the amount of fructose produced, as they have 1:1 ratio. Additionally, the concentration of fructose is directly proportional to the color intensity observed. And this creates a direct relationship between sucrose concentration and color intensity, allowing us to measure the sucrose concentration based on how intense the color is. They have provided a graph illustrating that the color intensity is directly proportional to the concentration of sucrose. Even if this direct relationship wasn't mentioned here, you should still be able to deduce it from the graph. And in this graph, the x-axis represents the concentration of sucrose and the y-axis shows the color intensity, and the graph forms a straight line indicating a direct proportionality between these two variables. And please keep remember, whenever you analyze a graph, it's essential to carefully examine both axes as they provided important information to you. And one key detail to note here is that the graph does not start at 0. There is a gap at the origin. This is important because it tells you that even when a small amount of sucrose is added, still the color intensity remains 0. So this suggests that the instrument may not be sensitive enough to detect very low concentration of sucrose, so it can't record any color change at such low levels. And let's explore another possible scenario to consider. Suppose your graph starts from a point on the y-axis rather than the origin, and this would mean that even when no sucrose solution is added, the sucrose solution is 0, but the graph shows some color intensity. So if this happens, it indicates that the machine detecting a color product even when there shouldn't be one. This can be like it says the machine has a baseline color reading or some contamination that is causing a false positive results. So understanding this concept is important because future questions might modify this scenario, testing your ability to interpret changes in graph behavior and what they could mean about the system's accuracy or sensitivity. Now we will look the questions one by one. I have already explained everything about the graph. The first question gives a reason why the color intensity is directly proportional to the concentration of sucrose. And you know, a direct relationship is because each sucrose molecule contains only one fructose. Therefore, as the concentration of sucrose increases, more fructose is produced according to the sucrose concentration, and so color intensity also increases. Therefore, you can show a direct proportionality. The next question is suggest why the line does not start at the origin of the graph. These things also I have already explained. You know that because the color intensity remains too low to measure accurately. And the last question, state why the concentration of maltose and lactose cannot be measured using this test. Initially, they have mentioned clearly that the chemical which is cause color change only can bind with fructose. Therefore, you can see maltose only contains 2 glucose molecules and lactose contains 1 glucose and 1 galactose. So both glucose and galactose cannot bind to the chemical and cause the color change. And next question is from May 2024. Here, the diagram shows two glucose molecules. Complete the diagram to show how these two molecules join by a glycosidic bond to form two products. So they have mentioned two products. It means the one is disaccharide and the other one is the formation of water molecule. They have already provided you two monosaccharides. So just you have to draw the glycosidic bond clearly, and then you have to show the H2O formation. For this, you have to focus on drawing the glycosidic bond correctly, and you have to make sure the rest of the molecular structure is correctly represented. And finally, you have to show the water molecule formed during the reaction. I hope this video was clear and informative. Thank you for watching and supporting my new channel. Don't forget to like, subscribe to Pearson Bio Academy, and share your thoughts in the comment section below. Stay tuned for part 2, where we will dive into the topic of polysaccharides. If you haven't subscribed yet, please make sure to hit that subscribe button so you don't miss out on future videos. Thanks again and see you in the next video.